All right. So welcome to the Intro to Confocal Microscopy, specific to Fluoview-1000 microscope that we have at the ICI up, down, and HSRB. So the first question when you do any kind of microscopy is to ask yourself, which is, what is the goal of microscopy? And it is to acquire fluorescent signal from cells or tissues which are either labeled with a fluorophore or your cells or tissue which are expressing any kind of fluorescent protein, and in turn to visualize the structure or structures to address any kind of biological questions, right? So the key over here is your tissues are labeled with a fluorophore or it's expressing any kind of fluorescent protein. So how do this fluorophores or fluorescent protein work? So that's represented nicely in this Jablonski diagram, in which the fluorophore, which is at the ground state, gets excited and it goes all the way up to the excited state. It cannot stay in this excited state for a long time. And the electrons have to come down back to the relaxed state. However, when they do so, they go farther in the wavelength, and while doing so, they also release the energy, okay, which results in fluorescence in a different spectra that we are used to seeing. So in this example, what you are seeing is you are exciting your fluorophore or fluorescent protein with a blue light, and this is typical for 488 nanometer wavelength, and that gives you green light. So this would be an example for FITC or for green fluorescent proteins. And the same thing can be visualized in terms of the fluorescent spectra for different fluorescent fluorophores or fluorescent protein. And what you see in this scenario is you have a broad excitation spectra as well as a broad emission spectra, right? So while we always say that I'm going to excite my fluorophore or fluorescent protein with 488 and I'm going to collect my emission at, let's say, 510 nanometer, which is green over here, you got to understand that there is a broad excitation and a broad emission spectra. So a tool like FP Base is very helpful in understanding the overlap between the fluorophores that you can select. And typically in fluorescence microscopy, you usually end up using four different fluorophores, right? Which is the blue, the green, the red, and the far red. And we know that these fluorophores um, don't have a huge amount of crosstalk. But now, if you try to put something like a CFP or a YFP over here, that will generate more of a crosstalk, and you have to be very careful about it. All right. So going back to the Fluoview-1000 for microscopy, it has got two different modes. One is the standard epifluorescence mode, and the second is a point scanning confocal mode. Now, when it comes to the epifluorescence mode on Fluoview, we are going to use this mode to check for fluorescence in the sample, as well as to get the specimen in focus. Ideally, I would suggest that when you bring your sample over here on the confocal, before you do so, you should check for fluorescence on the epifluorescence microscope that you have. I mean, now that you guys have Kians, you can check on that. This is just to make sure that you're not wasting your time all the way coming over here and then being disappointed and not seeing anything on the point scanning confocal mode. Does that make sense? Okay. So, and again, with the epifluorescence mode, you can check your sample into different modes. One is the transmitted light mode, and second is the fluorescence mode. Now, all of this is something which I will show to you when you are here in person. Will take about 5 minutes. But beyond that, everything that I will show to you on the confocal will be done remotely. So you will be sitting on one side, and I'll be sitting on the other side of the room, and I'll guide through things remotely. The other is the point scanning confocal mode. And this is where you will be acquiring your sample in the multicolor that we talked about, right? Which is blue, green, red, and far red. And we can, in fact, image things like a cyan or yellow fluorescent protein if need be. But I don't think your fluorophores are that complex that you need to do something like this. We can acquire multidimensional data sets, which are in X, Y, Z, as well as temporal dimension. Um, when it comes to temporal, we don't have a 37-degree incubator, so we cannot do that. However, if you just want to image something in time at room temperature, we can definitely do that. Your data sets will be quantifiable, and this is something which I was harping earlier to you, that um, when you acquire data, you want to make sure that your image settings are such that your fluorophore is not saturated, right? So um, you want to make sure that your data is quantifiable as such, and the data is publication quality. Okay, so the first thing to understand is the difference between epifluorescence versus confocal. So one of the common misconception with people is that when you come onto confocal, it gives you better resolution, which is fake news, I should say. You know, it's fake. You don't get better resolution on confocal. What you rather get is better contrast, right? So on the left, what you are seeing is epifluorescence, and on the right, what you are seeing is confocal. And confocal gives you a far more better contrast compared to what you are seeing on epifluorescence. So this is much more evident when you are trying to look at something which is very vesicular. It becomes it comes out as nice and green dots or structures in the sea of green that you are looking over here. Okay, so what is the difference between the epifluorescence and a confocal microscope? So in case of epifluorescence, as you might recollect from what we did on Friday, which is you have a light source, you have a bunch of different filter sets, which are changing depending on the fluorescence that you want to acquire. Your light source has got broad wavelength in it. So the typical spectrum ranges from 400 nanometers all the way to 720 nanometers. So that's what you are looking over here. You have light, which is all the way from 400 to 710 nanometers. You use an excitation filter to filter out the specific excitation that you want, which in this case, we are looking at a GFP or a FITSI based fluorophore. So we are selecting for the blue light, which is 488 nanometer. We have a dichroic mirror, right? So dichroic means two colors. It emits the light, um, which is the blue one or 488 nanometer onto the specimen, and it allows the light of longer wavelength to pass through, which is the green. Once it comes out of the specimen, it passes through the emission filter and gets detected by detector, which was on Friday when we looked at, it was a camera, right? So that's what is the structure of an epifluorescence microscope. In contrast to that, with a confocal microscope, we are using laser of a particular wavelength. Still, it has to pass through an excitation filter, which I'll talk later. It passes through the dichroic mirror, passes through the objective, and it excites the entire depth of the specimen. So for both the epifluorescence as well as the confocal microscope, you are exciting the entire depth of the specimen. Okay. Now, where does the difference between both of them come? Of course, the first is that you are using a particular laser, but the second difference that you are getting is in this mirror, and this mirror is going to scan the entire specimen the way you want, and you can also control the z depth that you want to acquire the image of. The light following the excitation of the sample is going to pass through back through the emission filter. But the key over here is we are not collecting the entire depth. We are eliminating any out of focus light using the pinhole aperture, right? So we are using a pinhole over here, which only allows us, let's say if our object of interest or plane of interest is this one, which is highlighted by pink. though we are exciting the sample in the other two planes, the fact that the pinhole can eliminate the out of focus light, which is this green one, gets filtered out, and we only get light which is from the plane right here, and that gets detected by the photomultiplier tube. And I'll talk about what photomultiplier tube does in a second, or in few minutes rather. Okay, so it's not
So,
PTM, it's PMT, photomultiplier tube.
and you said for the epifluorescence, there is no. Can you hear me? Right?
No, can you bring the microphone closer?
Yeah. Can you hear me now?
That's much better.
So you said that it's also the epifluorescence also does the depth, but doesn't.
Yes.
Okay.
So the light passes through the depth of the specimen for both the microscopy techniques.
But in the epi, it doesn't have the pinhole that uh, makes the other light go away. Kind of.
Exactly,
That's
exactly.
why you see this kind of
This
the
haze that
green being the haze with the green, I guess.
exactly
Okay. Thank
makes
you.
sense. Okay.
Hmm.
Now, while we are talking about this, I think for the sake of transparency, I should also say that there is another microscope in Clinic B, which is the multiphoton microscope. And the multiphoton, it only excites, shines the far red laser at a particular point in the specimen. So that gives you a better depth of your tissue, as well as you don't get any of this out of focus light. So you don't need a pinhole for a multiphoton microscope, just because the laser itself is a point source which goes and excites the specimen at a particular depth that you are looking for. Okay, so let's say in future, if you guys are doing some deep tissue imaging, if you have a tumor that you want to image, uh, I mean, if you guys work with liquid tumors, right? Or you also work with solid
Yeah,
tumors?
solid and liquid.
Okay, so if in solid tumors, if you ever want to visualize something deep inside, we can use a multiphoton microscope.
Mm.
Okay, so the key over here is that we are using the pinhole to eliminate out of focus light and just get the light which is in focus. So in nutshell, confocality helps in getting better contrast by eliminating out of focus light, which I think I've beaten down to death by now. And it also allows us to do something called axial or Z sectioning to obtain a nice three-dimensional image, which can be at times difficult with epifluorescence microscope. Okay? So just to run down real quick on the optical configuration of the Fluoview-1000. So this is like a cross section of how everything is spread out for Fluoview. We have got four different lasers: a 405, 473, a 559, and a 635. It's going to pass through this mirror, and the light will pass through the AOTF, which stands for acousto-optic tunable filter. Now the light that comes out of the laser is not exactly 405 nanometer in wavelength, and AOTF is going to fine tune that wavelength for you. It's going to pass through the fiber optic cables, through the mirrors, and this is where the magic of the point scanning happens, right? So we have got galvanometer scanning mirrors. What that means is, um, in contrast to epifluorescence, where you have a big bulk of light exciting the entire area, you have a laser point source, which is going to be smaller than the bulk of light coming through epifluorescence. So, how do you scan the point source of the laser onto different area of your specimen? So, for example, let's say um, if this is your area on which you are imaging your cell, uh, the point source is going to scan like this. Let's say if you have a 512 by 512 grid, right? So on a 512 by 512 grid, um, your point source will go through all of this 512 pixels in horizontal direction and 512 pixels in vertical directions. In order to do that, you have this galvanometer mirror, which helps this laser point source to travel on each of these pixels and keep on scanning. Okay, so the light passes onto your specimen, the specimen gets excited, it travels back. Again, you have the dichroic mirrors over here. The light passes through the confocal pinhole where the out of focus light gets illuminated. It passes through diffraction gratings, barrier filters into your photomultiplier tube, which does the analog to digital conversion of your image, and finally you get an image which could look like this. So that's in a nutshell the laser lines, the bunch of detectors that we have, and the key over here are the objectives. So we have a 10x and a 20x air objective. We have a 40x oil immersion lens, as well as a 60 and 100x oil immersion lens, and a water objective, a 40x water objective. So depending on your sample and the question that you are asking, you will be choosing different objectives. And this is something that I will cover much more in detail when you come up for training over here. Okay. So let me. This is always a pain to remove once I. Okay, here we go. All right. So when it comes to objectives, this is just a quick primer of what objectives are. They are not a simple piece of glass at the front and at the back. If you look at the cross section of objective, it contains lots of different lenses within. And the goal of this lenses is to make sure that the light which is collected from here comes out at one specific spot from the back focal plane of your objective. Okay, so when you look at the objective, there are a few different things written on them. For example, it says over here, it's plan apo, which essentially means it's corrected for flat fields. It's also corrected for aberrations, and there could be different kind of aberrations, which is a topic of different discussion itself. You have a 60x objective, it's 1.4, and this is the key number, which is the numerical aperture. And I'll talk about numerical aperture in next two slides. It's oil immersion lens, and another number which is important is 0.17, which is the thickness of the cover slip that the objective is optimized for, and the working distance, which is 0.21 millimeter or 210 micrometers total, and that's how deep your oil immersion lens, which is 60x, can image through your sample, right? So let's say if you bring a tissue slice which is 500 microns in thickness, and you want to image the entire tissue using a 60x oil, the point I'm trying to make over here is that you cannot do that just because the working distance of the objective, which is how deep it can image from the cover slip, from the bottom of the cover slip to the depth of the tissue, is 210 microns. So that's the best that you can get out of this. Okay. So lenses are complex and expensive. Use them, do not abuse them. Make sure you're not bumping or denting into them. Please wipe off the oil from the objective using lens paper, and something this is what I will show to you when you are here. Don't bathe the oil immersion objectives with unnecessary amount of oil. Typically, couple of drops of oil is pretty enough. And if the oil gets onto your air objectives, let me know. I'll clean that up. Don't hesitate in letting me know, just in case you do that by mistake. Because if somebody else is coming after you to use an air objective which has got oil, that will ruin the images for them. Okay. So when it comes to the NA is what determines the resolution of your objective. So when we look at the formula for NA, it's N times sine alpha, and N is the refractive index of the interface. So for example, for an air objective, the refractive index of air is 1, whereas for oil, the refractive index of oil typically will be 1.51 and some other number depending on the oil that we are using. So if you think about this, uh, the air objective is going to be further away from your specimen, whereas your oil immersion objective, because it's touching through the oil to the cover slip interface, is going to be closer to the cover slip. What that means is that there will be an angle formed, which is sine alpha, and that's going to be smaller for an air objective, and it's going to be larger for oil immersion lens. And this also means that the amount of light that you can collect through oil immersion lens is pretty high compared to the low numerical aperture objective. Okay, so if we look at this in action, what you see is, let's say if you want to use an objective of different numerical aperture and you want to resolve a bead like structure, which is a point size object that you want to resolve, how does the image look like? And how does the array disk pattern of a particular point source look like? So if you look at point 2, if you are using an objective which is point 2 NA, you get a lot of blurriness of your beads. And when we look at the array disk pattern, we see a bunch of array disks over here with a bright intensity in vertical axis. As we as we increase our numerical aperture, what you will see is you are now able to nicely resolve each of these bead-like structures, which you were unable to resolve earlier. And while you are able to resolve each of them, the array disk pattern also changes, right? So the array disk becomes thinner, which means you are able to resolve things much more nicely. So if we look at this diffraction patterns, let's say, for example, we are right here at the best numerical aperture. We are here. We are here somewhere with our kind of oh, that's interesting. Let's see if I can. Well, I give up on this. It won't let me. Well, you know what I'm trying to say over here, which is the pattern changes, right? So with lowest numerical aperture objective, the difference is this big when it comes um, to getting, when the difference comes to resolving the objects. Okay. Let me go back. All right. So again, resolution is the ability to differentiate between two distinct objects or two closely spaced array disks. And resolution can be defined by the formula, which is the wavelength of the light that you're using, divided by 2 times the numerical aperture, which can be derived from this formula, which is n times sine alpha. And the best resolution that can be achieved is 250 nanometer in XY direction, and 550 nanometer axially, which is in Z direction. Okay, so when we take a cross section of an object and try to resolve that, this is in XY direction, where if we uh, draw a line scan, this is what we see. These are two objects over here. They are below the resolution limit. And even if there are two objects that we know that they are present, they appear as one just because they are below the limit of resolution. Whereas in E, we can resolve the two objects and we can see two individual peaks for each of them. However, when we look at the same object in Z direction, we see an elongated structure, and that's because of the property of the light and the physics and the optics. You will never get a flat or isotropic resolution with what we are doing. Okay, so this is something to remember, that when you emit something in Z direction, you will always get this spread. And if you want to do a good Z sectioning, you have to collect enough Z stacks, and also you want to maintain the gap between each Z stack such that you can avoid this patterns when you do a 3D reconstruction. Okay, now when I said that you get this resolution, you only get this resolution when you are using the best objective and the best image acquisition settings. If you don't use that, you cannot get this good resolution. Okay, so now um, I want to again go back to this part, which is what's going on once you collect the light, which is at the photomultiplier tube level. What's going on? And when I talk about photomultiplier tube, I also want to touch onto two different areas, which I also touched on Friday with you, which is it's important to get a good image setting. And there are two different ways to do good image setting, aka increase the image brightness, and that is through laser power and through high voltage. When we were talking about epifluorescence, we were talking about um, reducing the amount of excitation light which was coming to make sure your sample is not getting bleached, and second is increasing the exposure of the camera, right, which is how long um, the camera shutter is going to be open to collect all the light. Now, those principles, it's kind of the same principle, but in very, very different ways. So, I mean, let me clear this and say it's not the same principle, but the idea is the same over here. Okay. So, what's the idea? The idea is the laser power, and second is high voltage, where the PMT comes into picture. So, when it comes to laser power, um, you can get a higher excitation of your sample if you increase the laser power. But at the same time, you will also bleach your sample because there's a limited fluorophore cycling time. So again, just like Friday, if you increase the laser power and your fluorophore is something like FITC, which is very light sensitive, it's going to get bleached and you won't be able to see a lot of it. Specifically, if you are using live cells and if you use a lot of laser power, it can make cells unhappy and kill them. Remember, laser also interacts with the nucleus, and nucleus is where the DNA is, and shining laser onto your cells can induce DNA damage, and this is much more relevant if you are using a low wavelength light like 4 or 5 nanometer, which is used to excite DAPI. Okay, so if you were to use DAPI for live cell imaging, your cells will be really very unhappy very fast and they will die because of DNA damage. Second is the high voltage. So this is where this is kind of a cross section of photomultiplier tube that I was talking about. All the light which is coming out of your sample goes and hits the outer layer of your PMT, which is the photocathode. The photocathode is going to convert the light into electrons. Your photomultiplier tube has got these structures called the dinodes. And what dinodes do is it amplifies the electron. So depending on the voltage that you are using, uh, the electrons will be amplified. Okay, so this is really important, which is this is what exactly we will be doing when we try to image our sample, which is try to fine tune the laser power and the high voltage. And once we increase the high voltage, we're going to amplify these electrons. And while the electrons are getting amplified, they are not specifically getting amplified based on the light coming from your sample, but they are also getting amplified from any kind of non-specific signal, which is also not part of your sample. Or it could be the background of your sample, right? So you got to make sure that you are amplifying not just the signal of your interest, but any of the noise also. And PMT also has its own noise, which I don't think should be a big deal for you, but something for you to know. Eventually, the electrons will be converted through all the electronics, which I won't talk about. But eventually, you have to convert this analog signal to digital signal, and that will be observed in the form of pixels, right? So that's what we are looking at. Um, in nutshell, images are nothing but pixels. So when we zoom into an image, you can either acquire an image on a confocal as 512 by 512 or 1024 by 1024. And remember, I told you that on confocal, what you are doing is you are point scanning every individual area. So every individual pixel is getting point scan. Your laser is in fact traveling. Your laser is in fact, let's see if I can get this work. Okay, your laser is in fact traveling through each of these spots and you are getting the image, right? So think about it. If you were to select a 512 by 512 image raster pattern versus a 1024 by 1024 image raster pattern, it's going to take much more longer to scan through a 512 a 1024 by 1024 image compared to 512 by 512, because here you have 3 times a larger area to scan compared to a 512 by 512 image, right? So two things will happen. One, it will take more time to scan 1024 by 1024 image, and second is the image is going to be big in size. I mean, the size of an image is no longer a problem, but definitely how long it takes is definitely something of your concern, which we will talk about in detail when I show you the sample on the microscope. Apart from that, when it comes to 1024 by 1024, the image quality is much more better. So let's say if you want to zoom in and show a structure of your interest, uh, a 1024 by 1024 will give you a better quality image. Uh, a caveat over here, the other way of looking at that is also by zooming in to your structure just when you start to image. So these are the kind of things I'll show you on the microscope. Now, if you're not happy with the resolution that you are getting on confocal microscope, we have something called structure illumination microscope, which can give you twice the resolution. So in XY direction, earlier we were getting 250 nanometers. With structured illumination microscopy, we got 120 nanometer resolution laterally and 250 nanometer resolution axially. So this is an example, a difference between confocal and structured illumination, where you can see some of the vesicles much more clearly between the two techniques. Okay, now if you're not happy with this resolution, we just got a STAD microscope, which I don't have a slide of, but we got a STAD microscope, and the STAD microscope can give you a resolution of 40 nanometers, right? So that's even better than this. So depending on the question that you are asking, we have got different tools. Now, one thing is the sample preparation for SIM is pretty much the similar one to that what you will be using for confocal, but sample preparation for STAD is a little different. So you cannot use the same antibodies that you are using for either confocal or for the SIM. Okay, so that's all I have for the theory. If you give me 5 minutes, I'll prep on the microscope and then we will jump onto the microscope side of things. Okay, so give me 5 minutes and I'll jump onto the microscope. Uh, so this is how, and can you hear me well? Can you hear me well?
Yes.
Good. Thank you. So this is how the screen will look like once you log in. So you will log in into the computer using your Emory ID. And once you log in, you will double click on the Fluoview software icon, which is right over here, which is all I logged on. And I'll create an account for you. And it's going to be your name, and the password will also be your name. Okay.
Mm.
The software will start. It will take about 30 or odd seconds for it to completely boot up. And it will make some clickety clackety sounds while it's connecting with the hardware. So while it's doing that, I'm going to go and put one of my slides. What you're seeing on the screen right now is like a little board. Let's think about it that it's a canvas, right? Canvas where a painter has to draw something and then put some colors into it. So this is your canvas where you are going to add the colors. Right now, it's already using the default setting, what I had the last time. But when you come in and create, and I will create your own settings, what you will see is this will be empty, this will be black. Okay. And the first goal is to fill this canvas with some colors. So before I do that, let me do one thing, which is change my objective. So you can change the objective from here. It's a 100x objective right now. I'm gonna go and change that to a 40x objective. I don't recommend you changing the objective manually. I would definitely use the software to control the objective. Okay?
Mm-hmm.
And I'm putting a couple of drops of oil right now. So the first thing you would do once you put your slide onto the stage of the microscope is to make sure that your sample is in focus. And to make sure that your sample is in focus, what you do is you click on the epifluorescence mode, right? So that's what I told you earlier when I was showing you the slides, which is the confocal has got two modes. One is the epifluorescence mode, and second is the point scanning confocal mode. So I'm going to enable or turn on the epifluorescence mode, which will allow me to see things through the um, binoculars. Okay, so I'm going to go in, adjust my sample fluorescence or the sample focus, and come back over here. All right, so now that my sample is in focus, the first thing to do is to adjust the colors, right? So this is where uh, the key comes into picture, which is how do we put in the colors? So to put in the colors or select the fluorophores, you can click on the die list, and I'm going to clear all of them right now and start from scratch. So I know that I have got three different fluorophores over here. One is DAPI, which is 405. Second is FITSI, which is 488. And third is FITSI, and I'll select 568 or 594. I know either of them works for me. So I'll go and select 568. Now, the green that I have is for actin. 568 I have is for mitochondria. Okay. So I'll be, and it's the same slide that we used last time. It's nothing different. I'm going to say apply to them. And the colors got changed over here too. And I'll close this. So for the moment, this is how it's, so when you do this for the first time, this is how it's going to look like, where you will have channel 1, which is 405, channel 2, which is 488, and channel 3, which is 568. And they are in three different groups, so group 1, 2, and 3. We can bring them in the same group, which means channel 1 and 2 will be imaged at the same time by the detector, right? So they are in the same group. But let's keep them separate for the moment. The second thing, when I told you earlier, was once we figure out the colors on our canvas, we have to figure out uh, the laser power, and we also have to figure out the high voltage. So you can control the laser power from here. Okay? So laser power for 405, 488, and 559. And you can control the high voltage. It says HV over here. And this is where the amplification of the electrons take place in the photomultiplier tube. Okay, so that is something which is controlled from here. All right. Uh, so let's start, and this is what I would do every time you are here. I will start with individual channels. Okay, so let's start with just blue channel to begin with. When you are ready to see your sample on confocal, you can hit focus times 4. Now, what you will see over here is you have a bunch of options: focus times 4, focus times 2, an XY repeat, and an XY image scan. What all of this means is the following. So if I draw this here, and we have, let's say, 512 by 512, and you know where I'm going with this. So assume this is 512 by 512 that you are having. when we are doing focus times 4, what it does is it scans the first region, it skips the second, and it skips the next 3 and images the fourth one. Okay, it skips the 3 and then images the fourth one. So focus times 4 means it images the fourth pixel area in the vertical direction, which means it's going to scan things very fast, but that would also mean that everything will be very pixelated. Make sense? Focus times 2 means it's going to scan every other. So it's going to scan this one, skip 2, and scan this. So it's going to skip every 2 and image the third one. Whereas XY repeat means it's just going to go and scan each of them continuously till you hit the stop button. Okay? So XY repeat means it keeps on scanning continuously till you go and hit the stop button. In contrast, for XY scan, it will just do a scan and then stop. Okay? So it depends on what you really want to do. You can select either of the options. And once for XY scan, it scans and stops, it gives you an option of saving the image. All right. So we'll be working with this 3 things. Try to look how it's going to look like. So let's just start with focus times 4. Once you hit focus times 4, you are switching all the everything to the point scan confocal mode. Okay, right now it looks very nice because the settings are good. So this is how it will typically look to you. Now, one thing is I selected 10% laser power and about 585 high voltage. That's been selected because I know it works the best for my sample. But typically, I would recommend to you that you should start with 10% laser power, about 600 high voltage, and then depending on the pixel intensity, you can change the laser power and change the high voltage. So let's see how the pixel intensity looks like. So right now it's all blue in color. Blue is very difficult to visualize for the eye. So what I would do is change this and bring up the LUT setting window. Now there are 2 windows which are very helpful. One is the, LUT setting window, which is this. And second is the information window, which is this, okay? So these 2 tabs are very helpful tabs, and I'll be using quite a bit of them. So with the LUT setting window that you got, the first thing I'll do is convert the blue color to grayscale. And over here, there is something called high-low. So this high-low button, what high-low will do is tell you whether or not your pixels are saturated. Okay, so let's have a look at this, how this is going to work out. So right now, we are looking at our sample. And if you look at the image, you can see the pixelation already. So if I zoom into it, the pixelation is very evident, right? And while it's pixelated, if I put my arrow on one of the pixels, you will see at the bottom over here where it says intensity, it gives you the intensity of each of the pixels, right, where it's highlighted. So if I put my cursor on individual pixels, it demonstrates, it changes the intensity value for each of these pixels. Now, uh, this is a 12-bit image, and we talked about bits last time. So this is a 12-bit image, which has got a bit depth from 0, or the pixel intensity values can range from 0 to 4095, right? So the number of shades of grays that it can have are from 0 to 4095. So if we enable the high-low lookup table, some of the pixels are, are becoming red in color, which means this pixel has an intensity value of 4095. If we go back to our dynamic range table over here, if I decrease the maximum value over here to, let's say, 3000, right? All the pixels which are higher than 3000 now will show red color. Okay, if I again reduce this to, let's say, 2 or 2500, all the pixels which are um, higher than 2500 will show red color. All right, so the high-low lookup table is a good way of knowing what is the kind of pixel intensity of your image or pixels, right? So depending on whatever value you select over here, this value also changes. All right. So I think we have beaten that down to that. So let's say if I'm seeing something which is pixelated over here and I want to reduce it, I will bring this down to about 500. Focus times 4. And now we can try to see what is the maximum value of one of our pixels. And the maximum value of one of our pixels is 1489, because this is when the pixel starts to become red. Make sense? Okay. So let's bring this up to about 550 and do a quick scan. Focus times 4. And if I increase my minima or increase my maxima, sorry, uh, my saturation of the pixel is at about 2605, right? So that's totally fine. We still have this entire range to fill up if we want to. So now we will do focus times 2 and see how it looks like. And we will do an XY repeat. Okay, so clearly the image looks a bit more better, and I'm happy with what I'm seeing. So I'll keep this image settings the way they are. I'll switch to 488 channel, and for 488 channel 2, I will use my settings at 600 for high voltage and about 10 for my laser power for 480. So keeping this, I'll again do a focus times 4 to see how it looks like. I cannot see anything, right? So I'll change my lookup table from green to gray. I'll try to decrease my maxima. So I'm decreasing my maxima. And what this is showing me is, well, I've got some signal. But what this tells me is the signal is pretty weak. And hence, I will increase my, well, I cannot increase much because I already have 600 as my high voltage. So I'll bring it up to 650, after which a lot of non-specific noise comes. So I'll keep it at 650 and do focus times 4. It looks a little better. But I know that I imaged this sample a lot, and that's why uh, it's looking like this. The sample has been bleached, which is what I know. So what I will do is I'll probably bring this up to about 10%. I can either go to a different field or we can just play with this so that you know how bad things can look. Okay. So let's just play with this area. So when we play with this area, right now the signal looks very terrible. What if I go to focus times 2? It looks a little better. What if I do XY repeat? It looks kind of better, but not a whole lot better. So one thing I'll do now is try to adjust my fine focus. Okay. So on this microscope, you can adjust coarse and fine focus on one dial itself. So I can change this to coarse and fine from here. So while I'm scanning this, I'll be making sure that my region is in focus, right? So you can see that I'm seeing increase in intensity while I'm changing my focus. So this is my right focal area or focal plane that I want to focus on. Let me just clear this, guys. So I'll stop it. And if I do an XY repeat now, woohoo, it looks much more better, right? So the problem was with the area or my focal plane. Once the focal plane got resolved, it looks much more better. Now this is actin, which means there is a nice structure to it. If you want to see this structure even better, there are two things you can do. One is you can increase the signal to noise ratio. So let's say if I zoom into this area, again, so there are two things you should know about the zoom, that you have a digital zoom. So this is the digital zoom, okay? And this is an actual zoom. So let's say, for example, if you have a camera and you physically go closer to somebody, right? You physically go closer to somebody. That's what this zoom button is doing. And let's say if on your phone, you are just pinching and enlarging something, that's what the digital zoom is doing for you over here, right? So right now I'm just zooming in. Right now I'm just zooming in so that you can appreciate the signal to noise ratio that I'm talking about. So things are very pixelated and there is some signal to noise ratio. So if you want to improve this, let's say what we can do is something called the Kalman averaging. So I have said I want to average what I'm imaging 4 times. What that means is the same pixel, uh, the same line that you are imaging will be getting imaged the same line. Let's say if you are imaging this, if you select Kalman averaging of 4, the microscope will scan the same line 4 times and then average all the pixel values that you get for the same point. Does that make sense? So it's gonna average everything that we are imaging here that we have imaged 4 times. Okay. So let's do that and see what we get. So I'm gonna do an XY repeat. And you can see the difference in the image, right? So let's do this again. This is with XY scan and with XY repeat or the Kalman averaging scan. And this is without Kalman averaging. You can see the difference over there, right? So all the noise that was coming out has been averaged out and the signal has now been averaged out and it looks much more nicer. The other thing you can do is, in fact, do an actual zoom onto your site. So I'm going to zoom into my sample. Let's say I zoom at like 1.7. And this is, if this is my area of focus, I can do an XY repeat. And now if I enable my Kalman averaging and do this again, looks much more nicer, right? So there are all these features of the software. Uh, at the end of the day, it's all about what is your question, right? So depending on the question that you have, the settings that you are using will change, okay? So if you ask me, tell me one setting which is kind of the magic wand over here, the answer is there is no one setting which is magic wand. Uh, the settings will change depending on the question that you have, okay? All right. So again, when it comes to saturation, this is not saturated at all. So that's what we are looking at. And let me go back. Uh, while we are at this point, this is kind of a good point to talk about a few things. You remember I told you that the other button which I like a lot is the information button, the I button over here. Now, if we click on the I button, the information button, it tells us a lot of great things. So it's telling us that right now we are imaging an area of 512 by 512, not an area, but uh, a region which is 187 by 187. And we are acquiring 512 by 512 pixels for that. Now, if I bring back my zoom to 1, now I'm imaging the maximum possible area, which is which is 318 by 318 microns. Okay, so I'm imaging 318 by 318 microns, whereas the number of pixels I'm acquiring are remaining the same. Now, if I change my zoom, what you will see is this area keeps on changing, right? But the pixels do not change. What this also tells you is how long it's going to take for you to image each of the pixel. So if you read the pixel time, it says it takes 2 microseconds to image 1 pixel. And what that means is how long your laser is far at 1 pixel. Okay? So your laser is far on 1 pixel for 2 microseconds. You can change this from here, right? So uh, you can change this and make it 4 microseconds per pixel, 8 microseconds per pixel, and so on. Now, if you change this, what you will also see is the total time that it takes to acquire 1 frame. So this is 1 frame. It's taking me 1.1 second to acquire the frame. If I increase the pixel dwell time, which is how long the laser is on 1 pixel, if I make it 4 microseconds per pixel, instead of 1.1 seconds, it's now 1.6 seconds, right? To image the whole frame. So let's assume we are only going to image everything using 2 microseconds per pixel. If I'm going to image everything at 2 microseconds per pixel and change a few of the other things, what you will see is, let's say if I end up putting the Kalman averaging mode on, right? So it's easy to do 5 times. So let's say without Kalman averaging mode on, it takes 1.1 second to image the whole frame. When I am averaging everything, I'm acquiring the same thing 5 times. If I'm acquiring the same thing 5 times, it takes 5 times longer. That makes sense, right? It's going to take 5 times longer. 1.1 times 5 is 5.5. So that's that. The other thing is, what if we, uh, I'll talk about a few of the things other time, but let's focus on the other parts. So if we look at the optical resolution, this is what the uh, the Olympus people provides, which is the best optical resolution that you can get is 200 nanometers. Okay. Right now, whatever settings we have, we are getting resolution of 621 nanometers per pixel. And uh, the our goal is to reach this resolution so that we can get the best of the resolution from the microscope. And the way to achieve that is by zooming in. So you can zoom in to the sample. And the other way of achieving this will be by increasing the aspect ratio. Okay, so right now we are imaging at 512 by 512. I can increase my aspect ratio to 1024 by 1024, and you will see that this is also changing. Okay, so let's go down to 1, and let's focus on this again now. So if I'm at 512 by 512 with Kalman averaging on, it takes 5.5 seconds. However, if I'm acquiring more pixels now, which is what I showed you earlier in the theory, if I acquire more pixels, now it takes 3 times longer. Instead of 5.5 seconds, it takes 16.23 seconds to acquire one frame. Okay, so let's see how it's going to look like. So let's compare the images with 512 by 512 and do an XY repeat over here. That's how the image looks like with 512 by 512. And now if I change this to 1024 by 1024 and do an XY repeat, you will first see is it's taking quite a bit longer. And that there isn't much difference in terms of how the image looks. But all of this will change if we try to zoom in to an area. So let's say if I want to zoom in into this area, it looks still much more nicer with 1024 by 1024 than I would do the same, let's say with 512 by 512. So let me go and do 512 by 512 and do an XY repeat. And now if I zoom in to this area, it looks much more pixelated. And again, when you are here on Wednesday, you can do all this kind of comparisons, save the image, compare, open on Fiji, so that you can actually see those differences. Okay. So are you good with the information window and what all these values mean?
Mm-hmm.
All right.
Yeah.
So now that you are good with all this, I'm going to get the settings up and running for the third channel. So once I'm here, I'll do my third channel, which is the mitochondria signal. Again, I've set it to 603, and my laser power is pretty high, so I'll bring it down to 10. And of course, it's not saturated, so that's pretty good. That's where I am. That's pretty nice. And the same thing with mitochondria. When you do focus times 4, looks bunch of pixels. Focus times 2, looks a bit okay. XY repeat. Now we are able to get the structure of the mitochondria. And if we do, let's say, And this was actually with Kalman averaging, right? So clearly it's not something that now it's with Kalman averaging. Um, we are able to see something, it's okay. But if you want to get mitochondria and the structures even nicer, we can zoom in to a particular cell and focus on that. So let's say, let's look at how it looks in the center and do an XY repeat. Okay, so looks a bit decent. Let's go over here. Okay, looks a bit decent. So now, if we change this to 1024 by 1024, because if I was to zoom in, right, it looks very pixelated. So if I go 1024 by 1024 and do a XY repeat, and now if I zoom in, it looks much better compared to before. It doesn't get very pixelated. Okay. So again, all these modes, it all depends on what is your question at the end of the day, right? Um, whether you want to see a structure which is very punctate. If you are looking at vesicle-like structures, you definitely want to zoom in manually, and then you also want to use a 1024 by 1024. If your goal is to have a nice publication quality image which goes into a paper, I would definitely image that using a 1024 by 1024, because then if you zoom in to a particular image, it won't get pixelated anymore. Okay.
Mm-hmm.
All right. So now that we have adjusted all the imaging parameters, let's take an image of all three channels. Okay, so now it's as simple as turning on 1, 2, and 3 up here and doing an XY repeat. And let me change this to this mode so you can go from multiple channels to single channel or single tile, and you will know what I'm talking about when I do XY repeat. So you get all three channels here now, versus if you only want to look at one, you can get all of them in one merge area. So let's keep it like this for the moment. I guess I did not like when I was changing things, so let it uh, get imaged once more. Oh, it was fine. All right, so it's imaging everything once more. And we can put color into our image. So we can go to channel 1 and say, I'll keep this grayscale. Go to my channel 2 and change that to, let's say, green, and I'll go to channel 3 and bring this to magenta. And if you want to look at all the merged channels, you can click on one of these windows. Let's say if I want to merge everything in this window, you can see the green border around the window. And once you see that green border around the window, you can turn on this, turn it on or off, and you will get this visualization. Okay.
Mm-hmm.
Similarly, if you want to put those two channels over here, you can bring them together and so on. Now, one thing that you will notice is you are seeing nuclei in the green channel, and that's not right. Something is different, right? Which means the question is your DAPI bleeding into your green channel, right? So well, that was a terrible one. Is your DAPI bleeding into your green channel? So If you want to know whether it's bleeding or not, what you can do is you can do something called sequential scan. Okay, so if I turn on the sequential scan, let's go and look at the information window again. So if you look at the time it takes to image the frame over here, it says 3.2 seconds, right? If I turn on sequential, what sequential will do is it's going to only scan my 405, then the laser for 488 will turn on, the 488 will get scanned, then my laser for 559 will turn on, and it will get scanned. Versus in non-sequential mode, all three lasers come up at the same time, and the images are acquired. Okay, so that increases the chances of crosstalk. So if I do sequential, because we are now acquiring each of them separately or individually, you can see that the time it takes to acquire the image also changes. With sequential on, it takes 9.7 seconds, which is 3 times longer compared to non-sequential, which is 3.2 seconds. Make sense?
Yep.
Okay. So let's image and see if this even makes difference. So the other thing you can do is in sequential, you know that the problem channels are the blue and the green. So you can put this into group 1, and that will increase the speed. Instead of, let's say, 9.7 seconds, if you put this into group 1, it's going to take 6.4 seconds. Make sense? So let's see how it actually works when we do a scan. So now that we are doing a scan, and let me turn on the Kalman averaging and do the scan so that we are comparing apples to apples. It's clear to you that you are no longer seeing the nuclei that you were seeing before, right?
Right. Yep.
In the green channel. So so this is one more thing for you to keep in mind, which is when to turn on sequential scan versus when to not turn on sequential scan. If you are imaging something and you see that you are getting co-localization, right? Which can be very exciting, which is like, oh, wow, you know, this is exactly what I want from my data. But I mean, co-localization is a very scary thing. You should be very careful about it. If your goal is to look at co-localization of something, I would always recommend that you turn on the sequential scan so that just in case there is ever a crosstalk between two channels, you don't misinterpret that data as a co-localization and actual signal. Okay?
Right. Is it always safer than to have the sequential on? I know that it increases the time.
Again.
Hmm?
Can you bring your microphone closer?
Oh, yeah. I was saying, is it always safer to then just put the sequential?
Yes, it's always safer to put on the sequential. But you know, if you are getting bored looking at how long it takes for things to scan, you can always play with your settings to see whether acquiring things in non-sequential also gives the same data as sequential. So when you acquire through non-sequential and you get the data wherein there is no crosstalk, then free and do non-sequential, and that will save you some time.
Right.
Because I mean, right now, if you think of it, it takes 32 seconds to image this much, right? And if you go to acquire Z series, it's gonna take forever. It gets very boring for you to sit over here and keep on staring at this. So, you know, the first thing is for any new sample to figure out the imaging conditions, optimize the imaging conditions. Spend like 15, 20 minutes optimizing all of that. And once you optimize those imaging conditions, life is good. All right. So now that we are we have a good image, uh, and let's kind of let me increase the dynamic range for that. Now it looks a bit nicer. Once we have this image, we want to save it. So what you do is you right click and save as. So there are two different ways. You can save this image that appeared during the XY repeat scan, or you can just do an XY scan, and that's going to acquire the image. And at the end of the image acquisition, it's going to ask you whether you want to save the image or not. So let's just let it just get finished scanning. So now it's asking us whether we want to save this image, and I'm going to do save as. We have a system on this computer to save the images, and the system is you go to the data drive D. Once you're in the data drive D, you click on old user data. That's just how our drive is configured. Go to 2020. Go down to Water Lab, and I'll create a folder for your name. And I would save the image over here, hello world. That's how people like to call the first thing nowadays. And you can change this from, one thing is, if at times you ever see a .oif image, that's not right. You should always save your image as .oib. Okay, so make sure it always gets saved as .oib and not .oif. And save this over here, and your image can be opened very easily using Fiji. So let's go to your folder.
So does it save all 3 channels?
Yeah, it does save all 3 channels. So go to Water Lab, and if I open this in Fiji, right, you get 1, 2, and 3. The funny thing is that it it does not put the lookup tables. You have to manually change the lookup tables, but that's the only pain. But you know how to change the lookup tables, so it's no big deal. So let's kind of just go and we can write a quick macro in Fiji that will let you change the lookup tables really quick instead of doing this manually every time. So it lets you do this. And there are some of the functions in Fiji in which you can make the structures look even smoother. Just want to kind of show that really quick. So for example, we can apply certain filters and still eliminate some of the noise so that the structure looks much more cleaner compared to before. Okay, so there are some of these options that you can always use in Fiji to make the structure look nicer. Okay, so I will cap it over here. There is one more thing I'll show you when you are here for the training, which is how to do Z-sectioning. And the other thing, which if you require, let's say we are right now only imaging 3 channels, but if you want to also acquire a 647 channel along with this. Well, let me just show that real quick, I mean, while we are doing this. Uh, so to do that, you will go back to Dilus and uh, enable something called the virtual channel scan. So when you enable this, it will do its own thing, open up a bunch of other windows, and give you a virtual channel controller window. Okay. So then it asks you in phase one what channels you want to use, in phase two what channels you want to use. So in phase one, I have used all these three channels. In phase two, now it's giving me an empty canvas to put colors in. Okay, and I will select what channel I want over here, and I will add 647 to my second phase and say apply. Okay. And once it's applied, I'm going to close this. And now I can adjust the, my parameter settings for 647. I mean, of course, you don't see anything because I don't have a 647 fluorophore. But assuming there was something, actually, let's do this. Along with 647, I can also, let me go back to my phase two. Let's say I also want to acquire 594. Okay, supply. So that's also going to give me some mitochondria. And I'll go back to my phase two. And it takes about 10 odd seconds to set everything up. And let's say I do this 600. So let's say if I do focus times 2, I get my mitochondria. And what you will see, interestingly, is the mitochondria is bleeding into 647, right? Because 594 and 647 are close enough for each other that my 594 signal is bleeding into 647. And if I turn on the sequential, that bleed through is gone.
Mm-hmm.
Okay.
Yeah.
So when you are having two uh, two fluorophores which are close to each other, uh, this can be a problem, and that's because of the detectors over here. But this is again a good example for you to know when to turn on sequential and when not to turn on sequential. Okay.
Mm-hmm.
So let me just acquire this image and see how it looks like. So I will turn the sequential on, my line average is on, and I'll just do, instead of doing XY repeat, if you want to acquire this, you have to go and say start over here, and that will acquire all 5 channels now for you. So it's going to acquire first 3.
So does it all acquire the XY or the XY repeat in this? Like
Sorry,
when you click
again?
on that start button, is it by the XY repeat or the XY?
No, the start is a capture. It's not XY repeat. It's just XY.
Okay. Oh, okay. So you already clicked that and you just capture it by that start button.
Yeah, so it's just capturing all the images.
Yeah.
So it's switching to phase 2, and now it will give you the 2 images of the second phase. All right, so at the end, we got a window with 5 channels now, right? Because we acquired 5 of them. We go back over here. So we got DAPI, FITSI, mitochondria, again mitochondria, and the slice, the 647, which was literally nothing. So let me actually, that's good. So in this way, you can acquire as many channels as you want to. But the key is spectral separation, making sure that there is no bleed through going on. All right. So that's pretty much it. You can save the image the same way that I showed you earlier. Save as and get done with it. Uh, so that should do it for the virtual part of the training. Hopefully, when you come on Wednesday, you will be much more familiar with the outlook of the software. It won't be intimidating for you and scary for you, so that you can just jump in. So what we will do on Wednesday is I'll show you how to put the slide, some of the hardware, and then you can put your slide over there. I'll be sitting on the other side of the room and monitoring everything that you are doing. Um, Hopefully, you should know how to work on things, but I will again go through things step by step. But it won't be brand new for you that you are looking at something for the first time, that you saw me things that you saw me doing things today. Okay,
Mm-hmm.
and I will send this recording to you in case you want to have a look at it before Wednesday.
Right. Okay. That was super
All
helpful.
right, cool.
Yeah.
And let me know if you have any questions. Okay.
Mm-hmm. Thank you so
All
much
right.
for
You're
your
welcome.
time.
I'll see you on Wednesday. Bye.
Yeah. See you.